for psychiatric disorders. But between all this, there's a whole lot of political and social issues that underlie how we diagnose and treat psychiatry. But today's talk is not about those aspects, but mainly about the biology and what do we do about understanding why these illnesses are caused. Since many of you are young students, this is perhaps a very important picture that you may have come across. This is the famous Solvay conference, Solvay meeting of 1927, which set out the rules for quantum physics and much of what we now take for granted. And 17 of the people in this photograph were awarded the Nobel Prize eventually over the next 20 or 30 years. Of these 17 who did get the Nobel Prize, 7 were known to have a mental illness or had a brother or a sibling or a child with mental illness. So this capacity of mental illness to actually have a whole range of possibilities of what the brain can do is very, very important to understand before we think of it purely as a disease. So that the idea that mental illness is only a disease, we need to dissociate ourselves from. It is not merely a disease, it's a kind of variation in the way the brain works. As you can see with this collage, authors, actors, generals, kings, musicians, surgeons, sports persons, everybody can have a mental illness. Anybody and everybody. It cuts across all groups, it cuts across all geographical locations, all racial groups, all possible cultural backgrounds. There is no society on earth which does not recognize the symptoms of mental illness. And it can obviously have very dramatic consequences. For example, in the case of Mad King George III, who was basically unable to rule for more than 10 years, to Stephen Fry, who's fated as a national treasure, to deaths like Praveen Babi or Virginia Woolf, or all the people on the right-hand side who all committed suicide. So it has a whole range of consequences. Why do we think it is so important? Because it is a very common disorder. This is a work done in Europe, and it shows that anxiety disorders, bipolar depression, all these disorders are very common in the population, several times more common than dementias, brain tumors, etc. But not only that, because they are so common and they require such extensive treatment, they are the most expensive of all disorders to treat. So addiction, anxiety, psychotic disorders, dementia, and mood disorders cumulatively take over almost 1/3 of all the disease cost in Europe, and a much larger fraction of the social cost, almost 40% of all the social cost, which in social distress in terms of how much time people have to spend and how protected the environments have to be made, how protective environments have to be made for these people, is very, very complex. We have very little understanding of the mechanisms of how this mental illness occurs, and there have been few advances over the past 50 years. So what is it that we try to do? We've tried to understand whether there's an inherited component to the disease of mental illness, and if so, to what extents. Can we identify gene variants? What are the environmental influences that could be used to explain these? And how do these combine and interact? Over the last quarter century, the Human Genome Project has been able to help us along this path quite a lot. We started towards the middle of the last century. By 1970s or 80s, we knew that these illnesses run in families. Schizophrenia, the major illness, is characterized by a period of delusions, hallucinations, disorganized speech lasting a month, and depression and mania by these other categories of being consistently depressed for 2 weeks in a non-reactive way, that you're depressed all the time, and mania being the opposite of 1 week period of intense happiness. Now, if you look at the way the slide is laid out, twins, monozygotic twins, almost have 50% chance of being ill. The first degree relatives have lower but significantly higher from the general population, between 6 and 17%. And it drops sharply down the curve, so that between the general population is 1%, and the further away you are from a relative genetically, the lower are the chances of getting the illness. suggesting that there is a genetic component to this. Obviously, there are devils in the detail. So between parent, sibling, and child and diazygotic twin, now technically a sibling and a diazygotic twin should be identical, but it is not, suggesting that the process of intrauterine growth or intrauterine problems or the environment of the uterus produces this difference, and more complex reasons why parents and children should differ. Similarly, with bipolar disorder also seems to run in families, and therefore we know that there is a genetic component to this. This is true. So if in the medical disorders, therefore we have rare disorders like Duchenne muscular dystrophy, hemophilia, phenylketonuria, where the genetics is typically a single gene, which is unifactorial, and there's a high recurrence rate in the family because these are Mendelian disorders. So if it is autosomal dominant, half the children have a chance of getting it, and if it's a recessive disorder, about a quarter of the children have a chance of getting it. So they have a high recurrence rate. On the other hand, these disorders are common disorders. Blood pressure, diabetes, schizophrenia affect 1% of the population. The genetics is quite complex. They are multifactorial. And in a given family, you may have only 1 out of 4 children getting affected, or 1 or 2 relatives in an extended family of 15 people affected. So it doesn't follow the typical Mendelian rules of inheritance. And therefore, it's much more difficult to find the genes that could be implicated. So what are the useful definitions? Single nucleotide variants. The human genome, of course, contains 6 billion pairs of DNA. 98% of that is intergenic. The human exome, what is expressed is about 2% of the entire 6 billion base pairs. It's around 22,000 pairs of genes, which not surprisingly are exactly the same number as in most vertebrates, right from mouse to us. So it's a fairly finite number of genes. About a third of the number of genes which are found in the common plants around us, the angiosperms. Of course, all of this will have to be expressed in RNA, and transcripts have to be made, and all that has to be done. Nucleotides and variants are, I guess by now all of you are familiar. A single nucleotide variant is a single change in either the G, CA, or AT, the nucleotides that make up the genome. And they're considered normal genetic variation if they are present in more than 1% of the population. So one of the methods that has been used is whether the pattern of distribution of markers which are scattered around the genome can differentiate between 2 sets of individuals. So that's a genome-wide association studies, and we'll be coming to that in a short while. Now, what do we aim for? Ultimately in psychiatric genetics, we need to better understand the language of life, and in the language of life, that of the mind itself. And therefore, we are trying to detect large deletions, frame shifts, or nonsense mutations. We are trying to correlate them with clinical, pathological, and molecular correlates. For that, we have to understand the population frequency. We have to understand the evolutionary conservation, the predicted biochemical effects, in vivo studies, correlate that with clinical information like diagnostics, prognostics, imaging, etc. And then combine them into case reports or series or all kinds of case control trials in the hope that ultimately this will become useful to the clinical bedside and perhaps even develop drugs for people who have particular kinds of genetic defects. The various kinds of mutations and how they change the language of life is put in this cartoonish kind of text on the right. So quick red fox jumped over the lazy brown dog. If the sentence just becomes quick lazy brown dog, it loses meaning. So that's a large deletion. A frame shift mutation, the word itself is shifted slightly. So red fox becomes red ox, so it's become a frame shift. And these become very nonsensical kinds. The quick red box jumped over the lazy brown dog. It becomes a complete nonsense statement. And otherwise, it becomes a kind of synonymous statement. So all kinds of things become very important to understand as to how is it actually going to change the function. The psychiatric or medical genomics is also complicated by the fact that there's a considerable amount of allelic pleiotropy. The same genotypes can give rise to very different phenotypes at different ages. So multiple phenotypes, multiple clinical aspects can be produced at different ages. A classical thing being sickle cell disease, which is common, for example, in Gujarat. And it can present as a fracture, it can present as splenomegaly, it can present with with multiple symptoms. But ultimately, the genetic defect is just sickle cell disease. And there can be lots of environmental influences that can also produce a disease state. For example, certain drugs of abuse, like long-term use of cocaine, can produce a schizophrenia-like symptom in anybody. And that may have nothing to do with the genotype at all. It is just a drug-induced state. So the environment itself can produce certain kinds of syndromes, and the genotype itself can produce various kinds of syndromes. This can be estimated by a simple idea that what is the relative risk of siblings, like we started out with. If one has a first degree relative with illness or psychiatric illness, risk to the sibling rises almost tenfold from that of the general population. And if you look at all the common diseases that we see now, major depression, alcoholism, nicotine dependence, ADHD, diabetes, Alzheimer's disease, they all seem to increase the heritability between 0.4 to 0.9. Schizophrenia and bipolar disorder slightly more, conduct disorder about this much, and diabetes is somewhere around here. So all these diseases run in families, and therefore the urge to understand how do these common disorders occur. So we have two major methods. One is that they can be tracked in a family using linkage analysis or the prevalence of a disease in a family. So you just do the genomic analysis and see what is common, which kind of genetic material is more likely to be shared in those who are affected in a family versus those who are unaffected. Obviously, to do that, you have to do lots of complex statistics because parent and children will share different kinds of genetic material, siblings will share different kinds, and these fractions have to be kept in mind. On the other hand, you can do an atheoretical manner based on the genetic variation, and you can do a genome-wide association studies in which you directly identify susceptible variants, and these can be used to develop novel biological insights or personalized medicine, which is the way a lot of the contemporary research is going. So linkage analysis basically relies on the fact that if the parents carry different kinds of markers on their chromosomes and this person is ill, then you can assume by the pattern of inheritance that because this person who shares a little bit of the yellow and the red derived from this person and who's the only other person who's ill, that somewhere along this whole fraction is the risk of the mental illness. So basically it just follows that pattern. And the most emphatic evidence of this is in this big family from Scotland, which was discovered almost 40 years ago. And this paper was published 20 years ago, in which because they were documenting cytogenetic anomalies in almost every child born in Scotland in the 60s, by the time they became adults in the 80s, they found that many of the children who had a particular kind of translocation between chromosome 1 and 11 developed a major psychiatric disorder. And when they tracked all these patients, they found that they were actually belonging to one large family. And they found that this translocation correlated with high risk of schizophrenia, with recurrent major depression, and with bipolar mode disorder. Whereas things like alcoholism, anxiety disorders did not seem to be linked to this. A large number of unaffected people, all these people also had this translocation, but they never developed any disorder at all. So, but it told you that the overwhelming proportion developed schizophrenia, bipolar, recurrent depressive disorder. And but many normals also did. So this became a probabilistic kind of observation that if you have this translocation, you are several times more likely, but not 100% definite, to have this disorder. So these kinds of studies have been done endlessly. For example, we have just published a series of 8 families in which multiple people are affected. As you can see, these are the children of these two patients. Between them have almost 10 people who are affected in the next generation. In this family, all the children are affected. This person was ill and all 6 children are affected. So this was analyzed using whole exome sequencing. Whole exome, as I mentioned earlier, covers about 2% of the genome, but contains all the 22,000 genes. And for contemporary medical understanding, as of now, we assume that most diseases are caused by some kind of a biological signal which varies, and that is obviously a protein function. So genes that carry exonic variants that are likely to affect proteins are likely to be so-called low-hanging fruit, especially in families in which so many people have a mental illness. So we found exonic variations in many of these genes. So our criteria was that you should detect a rare variant, it should be dysfunctional, it should predictably alter a protein, and it should be shared amongst all the affected individuals, because only then you can be sure, somewhat sure, that it causes the disease. So in all these families, we found a large number of variants. For example, the list is here, and many of them were completely novel. So these are the various genes that we found them in: the SETD6, LRRC8, NDRG, etc. Many of these genes are involved in various specific functions which are listed on the right-hand column, and they are all predicted to be highly damaging because they cause amino acid changes and they can cause fairly severe kind of defect in the protein function. Interestingly, most of them were confined to a particular family. They were not shared across families. So it turns out that almost every family may have its own unique risk for a mental illness, and this may not be shared across families. That obviously puts a challenge before us, because doctors diagnose illnesses by their similarities. And if people with different mutations are ultimately having a similar disease, how can we then reconcile these two facts? Either all these genes are part of some enormous super pathway that we don't really understand now, which we will in the course of time, but there seem to be multiple pathways to reach the final state of illness. Many of the genes that we discovered were many of the variants that we discovered were in genes that had already been picked up by. So that's approximately where we stopped. That the things that we discover are things that are being overlapped with leads that are being seen either through family studies or through GWAS studies, and they implicate processes like synaptic formation, memory consolidation, and are expressed in the brain, various parts of the brain that are important for mental illness. So therefore, we think that this could be at least some modest advance in understanding of psychosis. And overall, when you look at where these variants occur, we observe that genes that are involved in synaptic function, whether it's scaffolding proteins or vesicle machinery or cytoskeletal protein, or the receptors themselves or microtubule dynamics, many of them harbor genes that would alter the protein function and therefore alter synaptic function. They are all individually rare, but they all seem to be part of a network that seems to be plausible as a target for mental illness, for schizophrenia. On the other hand, we have these GWAS studies which rely on a relatively more complex model, which assumes that the susceptibility alleles for the disease arose many generations ago. These have now spread throughout the contemporary human population. New genetic causes have also started adding up, and there are many other environmental factors that can cause the disease. And the alleles themselves, because they have drifted over time, have incomplete penetrance, so they don't always cause the disease. So that the current diseases that you pick up in the clinic, the disease phenotype, will have a contribution from new alleles. It can have non-genetic factors. It may have people who have the gene but don't have a disease, and a majority of patients would have a particular kind of allele. That's what we hope. So there are two kinds of studies that have been used to do this. One is that you assume that you know something about the biology of the disease. You know that, for example, dopamine is involved in the risk for mental illness because all the drugs that you use to treat mental illness influence dopamine, and you look at everything that is involved in the dopamine receptor. It still requires testing hundreds and thousands or a million SNPs in the genome of all the possible pathways that dopamine would be synthesized and broken down in. And but that would still be a guess, because you're guessing a possible gene from what we know about mental illness. But since we do not actually know very much, these kinds of theoretical models have proved a bit inadequate. On the other hand, one can look at what is called linkage disequilibrium. You identify fragments of the genome which are shared between patients who are ill and those who are not. And if you study a large number of people from different parts of the world and identify which parts of the genome are shared in those who are ill, you will find a group of genes which are responsible. This may involve hundreds of thousands of genes, so we will still be in trouble, but at least you'll know that. So to go back to one of the earlier figures about heritability, then you find that these rare variants which occur in families which are transmitted are rare, very, very rare. They account for 0.0001% of the risk. And these are classically Mendelian disorders, things like chromosome 22q deletions, various kinds of reorganizations of these chromosomes, and neurexin genes which are seen to run in families. So these kinds of diseases are obviously very rare because they have to run in families, and they are hard to identify genetically because you need to have access to the particular family, and the individual person may or not be very informative by themselves. You may also have low frequency variants with intermediate penetrance, and these are the most variants which are identified by GWAS will be spread throughout the human population. They'll cumulatively or individually, each one of them will increase the risk only by a fraction, but cumulatively, they may explain a large part of the risk. So the issue is that do we have, when you want to make a meal for those who are fish eaters, would you want tens of small sardines, or do you want a big fish? Now, the big fish is obviously more difficult to get, but the small fish may be somewhat easier to catch. But obviously, some fish is better than no fish at all. So that's how we start doing this. Okay. Now, to do this, we obviously have to also look at variation and long-term evolution. We have to study how these genes have changed over time, and we have to study the genealogies of each population and long-term dynamics of the genome. Whatever it is, we find in the recent past, and this became qualified for an actual T-shirt, that you were able to find a gene which is implicated in schizophrenia at a possibility of 10 raised to minus 23. So this was a really interesting kind of observation, and you also found that these gene was shared across many, many disorders, of which more in the next slide. So, for example, the current studies now use 200,000 subjects, studied 1 million or more markers. And so it's only countries or societies that have such elaborate healthcare that can actually sample 500,000 people at a time for mental illness. And when you sample 500,000 people, controls and patients combined, you get a large number of genes that are contributing to the risk of illness. Not only do you get genes, not only do you get variants that contribute to disease, you find that these syndromes of autistic spectrum disorders, bipolar disorder, major depression, and schizophrenia actually have somewhat shared characteristics. So that bipolar disorder and schizophrenia have many things in common, as you can see in these. And these are all expressed. Many of these genes that are being picked up as high level of confidence are expressed predominantly in the cortex and not in the liver. So not only do you have genes that you're discovering genes that are being variably expressed in the brain. Now this is a big advance, because at least you know that you're not shooting in the dark anymore. You're discovering genes which are changing the way the brain works, and which then also causes brain disease. At least you can make that judgment, which was not possible till now. So what are the benefits of this? The benefit is that it is absolutely atheoretical and data-driven, which, as all of you are engineering and science students, is very, very advantageous. You cannot have a biased experiment. It is much better to let the data talk to you, and your hypothesis should follow the data rather than the other way around. So because it is atheoretical, because there is no a priori hypothesis, our understanding is likely to be more, inverted commas, genuine. This data can be mined over time. So you do 500,000 people today, 500,000 people 2 years later, all this data can be made cumulative. And this can become a very large body of confirmatory evidence. We also are able to rule out any one gene for mental illness, which is very, very important for us to understand. We can map ancestries and ethnicities. Many of these signals disappear in populations which are non-European. And therefore, what does that actually mean? We also need to understand. Okay. So we need to understand that these variants that we are picking up will identify at least some of the risk factors that are contributing to disease, but not the ones which are very small contributors as yet, given the sample sizes that we have. Okay. Like I said, if you compare these risk factors between East Asian and European population, you find that these alleles, that the genes actually differ a little bit because the genes themselves are different in different population. And they differ by a fragment of a recipe between 0.4 and 0.5. But if you compensate for that difference, you find at least the same genes are involved in both East Asian and European populations. So not only are you able to compensate for genetic variation across the races of man, because we know that there is huge amount of diversity in human beings, but overall, the risk genes seem to be somewhat similar. They may differ in their minute detail, but overall, we are finding the same kinds of genes being implicated in the risk of mental illness. But this has been possible only when we have started combining populations again of almost 500,000 each across populations. To give you an example, these are two of the genes that have been identified. So one of them, which is least variant across populations, the potassium channel gene, which is approximately the same ratio of its presence in American, African, European, South Asian, and East Asian populations. As you can see, it's present in almost 30% of the population and seems to add a little bit to the risk. On the other hand, one of the other alleles which has been picked up is this gene in the PPR23A, which controls cell division, which varies enormously between American, African, European, Asian, South Asian, and East Asian population. So you get a huge range of variation. And each time you look at the impact of this variant in different populations, you would need to compensate for this difference. In India itself, this is a study done in collaboration with colleagues in Delhi in the Institute of Genomics. So these are variants in genes that are involved in the synthesis and breakdown, in the breakdown and the action of dopamine. And you see that there are clusters of these variants. So in all these variants these variants that we are detecting are much the same as in the proportion of these is much the same as in European. And to some extent and they are much rarer in the Japanese and in the Yoruba, but they are almost the same proportion as in the European population, and these are in this segment of India. Whereas others, which are almost absent in the European and very rarely present in the Han Chinese and Japanese, you find them towards the eastern belt of India. So even geography seems to play a role in the way we handle dopamine and various neurotransmitters in the brain. So ultimately, what is it that we have come up with? Almost 1,000 loci in the genome may contribute to risk of schizophrenia. About 3/4 of them may be involved, maybe near a protein coding gene, and some of them may be altering other kinds of genetic pathways that we don't know about. Most of them, however, are in intronic regions. And therefore, we feel that the impression is growing that they contribute to disease by altering gene expression rather than protein structure. Then how will we study this? So over the last 10 years, all the tools of modern science have been used to understand the genetics of mental illness. You have experiments using whole exome sequencing, genome sequencing, transcriptome analysis, copy number variant analysis, and high throughput methods of various kinds. And for each of them, you've been able to find some aspect of schizophrenia biology. Most of this is leading to pathways that we don't know as yet. So there are changes in the methylome, there are changes in the cytoskeleton, there are changes in cell-to-cell adhesion. These are processes that we have very, very poor understanding of. So we need to go back to the drawing board. We need to go back to the bench and understand what is the biological impact of these variants. So some of this work is now possible. So people have come. There is something called the Stanley Brain Foundation in America, which invites people to donate their brains for study, and they have a collection of several hundred brains, autopsied brains from patients with bipolar disorder and schizophrenia, and they find that many of the GWAS signals that we are detecting are overrepresented as being differentially expressed in brains from patients who have bipolar disorder or schizophrenia. The vast majority, of course, are all shared across everybody, but there are certain which are shared between these two disorders and certain which are unique, pointing out that not only are you detecting genetic variants, you're also detecting the downstream effects of the genetic variants, and the fact that the brains of these patients have different kinds of gene expression patterns. And most of the genes that we are detecting if you take all the protein coding genes in the human genome, the number of genes that you detect to have functional variants are being associated with neurodevelopmental disease, autism, epilepsy, or schizophrenias, they all come out as having more likely to have damaging genes, damaging variants. This raises a very important evolutionary problem or biological problem, because as many of you will know, since you are much younger and you therefore will remember your early life, that birth is actually a very, 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 very statistically improbable occurrence. A large number of the millions of sperms and hundreds of ova, only one needs to be fertilized. Of the several 100,000 fertilizations possible, only one survives the first few weeks of pregnancy. Most of them are filtered by the biological processing. So all mutation, all variants are weeded out, and only a few survive the first 3 months. And only after that is healthy survival possible. So genes which do not impact fetal development will pass this filter. But because human beings are not designed to live for 50, 60 years, as the persons grow older, many of the genes which have escaped this filter, because they don't actually affect fetal development or childhood, will pass this quality check. So psychiatric disorders lie in this segment, that these genes do not affect the whole survival as such. There are subtle changes in behavior, and therefore they pass the quality control of pregnancy itself. What do we do with all this? Now, the reason why I chose the title is because this paper was published, The Maddening Hunt for Madness Genes, 25 years ago. And I thought, since most of you are well below 25, this talk would be a summary of this. The maddening hunt for genes that influence susceptibility to mental disorders may become less maddening through the parallel progression of human and animal research on the genetic basis of complex behavior. So what is it that is being done now to reduce this effect? So once we find the gene for the disorder, you can culture neurons. You can culture neurons from LCLs and make sure that they have the variant that you're interested in, or insert that variant with CRISPR. You can then study what is changed in the expression pattern of that. You can then figure out that in response to immune parameters, what happens to the neuron. Turns out in the presence of inflammatory markers, this variant alters the kind of synaptic pruning that goes on. Synaptic pruning is critical because schizophrenia or mental illness typically occurs between the ages of 15 to 25. That is the age in which your brain is actually cutting back its connections. As you all know, you learn trigonometry and algebra around 12 or 13 years of age. Your brain sprouts new connections. You have the capacity to understand algebra, music, fashion, interesting conversations. And by the time you're 25, you're narrowing down your interests. That's when synaptic pruning is occurring. Something in that synaptic pruning or synaptic biology is supposed to get altered in mental illness, and 90% of those who develop a mental illness will do so between the ages of 15 and 25. So the fact that we could discover something involved in synaptic pruning was very, very interesting. However, all this was done only in the European. This particular gene seemed to be at risk only in the European population and was not a risk factor in the Chinese or the African populations. So whatever it is, we found this mechanism, but is this the only mechanism? We have no way of knowing. So to go back to the beginning, since you're all undergraduates, as you know, this famous statement by Dobzhansky, nothing in biology makes sense except in the light of evolution. And medicine is applied biology, and therefore biology should be considered evolution in action. Because we share the genome and the brain across species, this commonality therefore makes science possible. And the differences between human beings and across species make science necessary. Why is this important? Because the brain evolved, let's say, in certain aspects of human beings. This arrow is displaced, so you can place it somewhere different now. Where is it gone? The arrow has got displaced because the slide orientation has changed. But anyway, you understand what I mean. So of all the animals, we are somewhere along this region as one of the various species of animals. As a neural process, dynamic consciousness of which is altered, for example, when you have mental illness, arose in reptiles around 250 million years ago. And as you can see in this, from the fish to the human beings, our neocortex and archicortex ratios have deviated so that more complex brains have now become possible. But these brains are still there, or parts of a similar kind of process has occurred in both fish and reptiles. So it's not as if we are the only brains. Animals like octopus have distributed brains, which work just as well. They also learn, they also move, they have complex perception, but the concepts of perception and memory may be completely different in the octopus brain. Of course, we think of human beings as being on top of this, but actually there is an evolutionary trend which is shared across things. So what is it that we would have to do? We would have to understand these genes at a molecular level, at a cellular level, and at a circuit level, because we need to understand how this is occurring over time. And since biology is shared, we can do modeling across scales. If we have a DNA change, we should be able to study the chromatin, the RNA, the protein, the cell, the circuit, the network, and finally the behavior. None of these may be straightforward. And you may have to therefore use everything possible, from brain organoids to primate studies, to rodent models, to zebrafish, fruit fly, elegans, and cell culture. So at our lab in NIMHANS, we are in the process of developing brain organoids, cell cultures, and doing at least some of this work. In collaboration with NCBS, we can do some aspects of this, and I'll be telling you something about these. So from the genes that we derive from WGS or GWAS, we still have to understand how they actually change the transcriptome, how they change the structure of the chromatin itself, and then finally influence cellular behavior. So this is an elegant experiment which was done a few years ago, I think 2 years ago, in which they inserted many of the genes that are implicated in the risk of schizophrenia into a zebrafish model, 30 of them, or 132 of them, and about a quarter of them turned out to produce changes in the zebrafish brain and in the behavior, which was similar to that produced in human beings. Now, this was an inconceivable concept a few years ago, that you could actually be able to mimic a complex disease like schizophrenia in a zebrafish model. But ultimately, it turns out that you can study the development of the forebrain, you can understand the social behavior and the way the zebrafish handles social stimuli in much the same way as you understand patients. And a quarter of the genes that were observed for schizophrenia risk were found to produce a phenotype which was quite understandable in zebrafish. In our own lab, we are trying to grow cells from patients and obviously controls. So cells have, nature does not like a vacuum. So if you grow a cell culture, make a kind of a valley in between, over time, cells from both sides migrate to fill up the gap, so that in 15 hours or so, the gap has more or less disappeared. So these are the control lines, and these are lines derived from a patient, cell lines derived from a patient. And then you can see there is a slight difference, perhaps not very detectable on the naked eye, but there is a slight difference in this. What is more important, what is more startling, is the fact that cells from patients go back and forth in a fairly linear manner, as you can see with this blue line and in the green line on the other side. whereas those from the patients go all over the place. They are not following a similar, as efficient, a straightforward path. So that they spend too much, they travel technically a longer distance, but as far as the destination is concerned, they have traveled a shorter distance. So whether there's an actual basic cell change, not only in the behavior of the person, but in the behavior of every cell of the person. So you can imagine how mental illness is now being imagined as a conceptual disorder of the individual cell as well as of the individual psyche. An example of some other work that is being done, for example, in IIT Chennai, one of the genes that we picked up in those families was something called the LRRC8B, which controls sodium, the electrolyte composition and the water composition of a cell. It controls the water and solutes and sodium through its action on calcium signaling and the action of this particular pore. Now, this particular thing, when it is damaged, it disrupts it and changes its firing pattern. So, Amar Kanti Bera at IIT Madras is now culturing cells from these patients with the hope that we will be able to then go back and figure out why the cells from these patients behave differently. Is it a function mainly of electrolyte balance or maintaining excitability of the cell membrane and therefore of the brain membranes? So we would be able to go back and forth between the actual patient and the cell biology. So what is it that we have finally got at the end of this quarter century? We've got hundreds of small hits. There is no big game out there. So we have to change our very perspective of psychiatric illness. So at the end of this, we have reached a milestone that at least we understand 1 to 2% of the genetic risk for mental illness. But as you know, in science, every question gives rise to more questions. We may be able to detect multiple genes of small effect, and thousands of genes may be involved, but we still have to unravel how they express, how they interact, how they are regulated, and how they are changed in the brain. And for this, we need science and engineering backgrounds, because unless we really understand how all this changes in the brain and all the various processes, right from the biology of the membranes and the neurons and the circuits, but basically how does it translate into information processing? How does it translate to decision making? How does it relate to the structures of the brain? Can we detect changes in the structure using MRI or functional MRI or whatever? And all this will be necessary to understand what is the nature of the human brain and what is the nature of the human mind, which is ultimately what we are interested in. So we have traveled far, but we always, as scientists, are skeptic that are we on the right road? Of course, there may be no differences. Like in the first slide, the participants of the Solvay conference, now would you call them patients or would you call them geniuses? Is there any difference between the seven out of the 17 who got the Nobel Prize and the others, and who developed a mental illness and the others who didn't? So how do we define disease in itself? There is no acceptable way of defining a psychiatric disease which is universally applicable. By universe means, I mean all times and all spaces. A person may become ill, may become all right after 2 years. A person may be highly creative and develop a mental illness after 5 years. So we have no way. The genes are constant right through. Why are we thinking the genes have caused only the disease? They may also have caused the creativity. They may also have caused the other ways in which the person has lived his life, his or her life. So we need to understand that disease is a transitory thing, whereas genes and the lifespan is a very different thing. It's quite likely that the biological and the genetic reasons for developing a disease are too subtle and specific to be analyzed by existing technologies. They may involve particular groups of cells or particular pathways or subtle network issues that we don't even begin to understand. Of course, population differences may obscure all this because the differences are even so subtle that the larger differences between populations can obscure this little variation. And the change may be too complex. After all, we are all aware that human brains are perhaps slightly more complex and have gone through a very significant change over the recent past, because the human cultural history of last 3,000 years has produced technologies which have changed the way our brains work. The human population was less than a billion 200 years ago and is now 6 to 7 billion. Most other large vertebrates have been wiped out. So we've become a single vertebrate brain, which is very complex. And this is the interface, the interaction between these large complex brains, which are crowding the earth, is simply too complex to be understood by a genetic snapshot. So so I'm ending with a note of skepticism but ultimately these are all things that we need to understand, and we really need young people to come in. It is a bit of a challenging field, but it's also the fun of it, because it is really the island of medical ignorance. We have no coherent scientific model to understand brain disease. Why does epilepsy occur? Why does dementia occur? Why does schizophrenia occur? And we need to discover these models, because 1 in 3 families will have somebody with a mental illness. And to keep on doing stopgap or band-aid treatments like what we do now is simply not good enough. So we need to discover more about these diseases so that we can help patients live their lives better. Thank you for your attention. The work, of course, has been made possible by intensive collaboration between the NCBS, INSTEM, and many, many colleagues at NIMHANS. And this is a snapshot of our molecular biology lab, which is what I've been talking about. And just to reemphasize, the mind is the gap that we have in our knowledge right now. Thank you for your attention.